Gametogenesis is the process by which male and female sex cells or gametes, that is sperms and ova, are formed respectively in the male and female gonads, that is testes and ovaries. The gametes differ from the somatic cells of the body. Their nuclei contain only half the number of chromosomes. Meiosis forms the most significant part of gametogenesis. Spermatogenesis is the process of sperm cell or spermatozoa development in the male gonads called the testes. It comes from the Greek word sperma, meaning sperm or seed, and genesis, meaning origin. In the vertebrates and insects, the testes are composite organs consisting of numerous seminiferous tubules. In vertebrates, early stages of cells called spermatogonia are located at the inner surface of the tubules in an epithelium-like arrangement next to the basement membrane, whereas in the later stages of differentiation, including ripe spermatozoa, it lies nearer to the lumen of the tubules. Rounded immature sperm cells undergo successive mitotic and meiotic divisions called spermatocytogenesis and metamorphic changes called spermiogenesis to produce spermatozoa. Sperm cells are continually being produced by the testes, but not all areas of the seminiferous tubules produce sperm cells at the same time. One immature germ cell takes as long as 74 days to reach final maturation, and during this growth process, there are intermediate resting phases. Testis. The testis is a compound tubular gland enclosed in a thick fibrous capsule called the tunica albuginea. Numerous seminiferous tubules lie in a bed of loose connective tissue called the stroma. Each seminiferous tubule is surrounded by a fibrous sheet of connective tissue. It is lined by a specialized stratified epithelium which forms the germinal epithelium. In the adult, two kinds of cells are present in this epithelium: the Sertoli or sustentacular cells and the germinal cells. The Sertoli or sustentacular cells. Sertoli cells are the epithelial supporting cells of the seminiferous tubules. They are derived from the epithelial sex cords of the developing gonads. They are tall, simple, columnar cells which span from the basement membrane to the lumen. They surround the proliferating and differentiating germ cells, forming pockets around these cells, providing nutrients and phagocytizing excess spermatid cytoplasm, which are not needed in forming the spermatozoa. They are connected to each other by continuous tight junctions that seal the tubule into two compartments, the basal and adluminal. Large molecules cannot pass between the basal and adluminal compartments. This is called the blood-testis barrier. Sertoli cells support the germ cell progenitors and help to transfer nutrients from the nearby capillaries. The developing spermatogonia rely on the Sertoli cells for all their nourishment. Sertoli cells also produce testicular fluid, including a protein that binds to and concentrates testosterone, which is essential for the development of the spermatozoa. They also help to translocate the differentiating cells to the lumen and phagocytose degenerating germ cells and surplus cytoplasm remaining from spermiogenesis. The germinal cells. The germ cells or sex cells make up a stratified layer of 4 to 8 cells deep. The cells differentiate progressively from periphery to lumen in the tubule. Next to the basal lamina lie the immature cells, spermatogonia, all derived from cell called stem cells in the outer wall of the seminiferous tubules. The stem cells are composed almost entirely of nuclear material which begins mitosis. Half of the new cells from this initial crop go on to become the future sperm cells, and the other half remain as stem cells so that there is a constant source of additional germ cells. Spermatogonia destined to develop into mature sperm cells are known as primary sperm cells. These move from the outer portion of the seminiferous tubule to a more central location and attach themselves around the Sertoli cells. In the course of spermatogenesis, the primary germ cells move towards the lumen from the outer portion of the seminiferous tubule and attach themselves around the Sertoli cells. The primary sperm cells then develop somewhat by increasing the amount of cytoplasm and organelles within the cytoplasm. After the resting phase, the primary cells divide into secondary sperm cell, which now contains half the initial number of chromosomes. The following developmental stages are thereby passed through during spermatogenesis: A spermatogonium, B spermatogonium, primary spermatocyte or spermatocyte order 1, secondary spermatocyte or spermatocyte order 2, spermatid, sperm cell or spermatozoon. The process of spermatogenesis can be subdivided into two successive sections. The first comprises the formation of secondary spermatocyte from spermatogonium and is termed spermatocytogenesis. The second one comprises the differentiation or maturation of the sperm cell starting with the spermatid phase and is termed spermiogenesis or spermiohistogenesis. It includes the following phases. A. Multiplication phase. Among the spermatogonia numbering over 1 billion in both testicles, several types can be distinguished. At sexual maturity, the undifferentiated primordial germ cells divide several times by mitosis to produce large number of diploid spermatogonia, which are of two types, type A spermatogonia and type B spermatogonia. Type A spermatogonia serves as the stem cells which divide to form additional spermatogonia. Type B spermatogonia are the precursors of sperms. Certain type A spermatogonia cells divide mitotically and reproduce themselves, by which the spermatogonia population is maintained. The beginning of spermatogenesis is initiated by the daughter cells, which are second group of type A cells that remain bound together by thin bridges of cytoplasm. By the preservation of these cytoplasmic connections, spermatogonia are inducted into the spermatogenesis process. After further mitotic division, type B spermatogonia are formed that also divide themselves mitotically into primary spermatocytes. These cells now in the pre-leptotin phase of the first meiosis enter the S phase. They double their DNA content, leave the basal compartment and reach the spatial luminal compartment. After the S phase, these cells pass through all the stages of prophase, which lasts about 24 days. The long prophase is followed by metaphase, anaphase and telophase. Growth phase. Each type B spermatogonium actively grows to a larger primary spermatocyte by obtaining nourishment from the nursing cells or Sertoli cells. C. Maturation phase. Each primary spermatocyte undergoes two successive divisions called maturation divisions. The first maturation division is reductional or meiotic 1. Hence, the primary spermatocyte divides into two haploid daughter cells called secondary spermatocytes. Both the secondary spermatocytes go directly into the second maturation division, which is equational or meiotic II, out of which the spermatids emerge. Since in the secondary spermatocytes neither DNA duplication nor a recombination of the genetic material occurs, the second meiosis can take place quickly. The two haploid spermatids contain only half the original DNA content. Thus, 4 haploid spermatids are formed by each primary spermatocyte. The secondary sperms further undergo maturation before it can fertilize. The transformation of spermatids into spermatozoa is called spermiogenesis or spermateliosis. It corresponds to the final part of spermatogenesis. The motile and active spermatozoa are later on known as sperms. Thus, 4 sperms are formed from one spermatogonium. This process lasts several weeks before they are transformed into sperm cells with the active assistance of the Sertoli cells. The nuclear material condenses into oval shape and develops as the head of the sperm. The head is covered partially by a cap called the acrosome, which is important in helping the sperm to gain entry into the egg. To the opposite end of the head is the tailpiece, derived from the secondary sperm cell's cytoplasm. The following transformation changes takes place during this process: nuclear condensation, acrosome formation, flagellum formation, and cytoplasmic reduction. Nuclear condensation. The nucleus becomes smaller, denser, and flattened, along with condensation of the nuclear contents. The nucleus appears oval from above, and from narrow side it appears pear-shaped. The acrosome lies over the tip. Nucleus and acrosome form the sperm cell's head that is bound to the midpiece by short neck. Acrosome formation. Here, the cap or acrosome forms. It contains enzymes that play an important role in the penetration through the zona pellucida of the oocyte. The Golgi complex produce the vesicles, which then merge to a larger formation that settles close to the cell's nucleus and finally inverts itself like a cap over the largest part of the nucleus. The acrosome corresponds functionally to a lysosome and thus contains lysosomal enzymes like proteolytic hyaluronidase, among others, which are popularly known as sperm lysins. These are used to contact and penetrate the egg or ovum at the time of fertilization. Flagellum formation. In this stage, the sperm cell tail is generated. The future axonomal structure grows out of the distal centriole. This consists of a bundle of 9 peripheral double microtubules and 2 single ones in the center. During its development through the rotation of the nucleus and acrosomal vesicle, the flagellum primordium comes to lie on the opposite side of the acrosome. 4 parts of the finished flagellum can be distinguished: the neck, which contains 2 centrioles, proximal and distal, among other things. The midpiece consists of sheath of ring-shaped mitochondria grouped around the axoneme to provide energy for the flagellar movement. The principal piece has a sheath of ring fibers around the axoneme. The tail consists only the 9+2 structure of axoneme. The mature sperm cell is approximately 60 mm long and completely enveloped by the plasma membrane. Cytoplasmic reduction. The cytoplasm of the spermatids that is no longer needed is phagocytized by Sertoli cells or is disposed of in the lumen of the tubules. The sperms thus formed are microscopic and motile cells. During sperm cell production, considerably individual variations exist that are also partially influenced by psychological factors. Roughly 100 million sperm cells are produced per day. In each ejaculation, an average of 50 to 200 million sperm cells may be present. They remain alive and retain their ability to fertilize an ovum within 24 to 48 hours after being released in the female genital tract. A typical mammalian sperm consists of a head, neck, middle piece and tail. Head. It contains anterior small acrosome and posterior large nucleus. Acrosome is formed from Golgi body of the spermatid. Neck. It is very short and is present between the head and middle piece. It contains the proximal centriole towards the nucleus, which plays a role in first cleavage of the zygote, and the distal centriole, which gives rise to the axial filament of the sperm. Middle piece. The middle piece of human sperm contains the mitochondria coiled round the axial filament called mitochondrial spiral. They provide energy for the movement of the sperm. So, it is the powerhouse of the sperm. At the end of the middle piece, there is a ring centriole with unknown function. Posterior half of nucleus, neck and middle piece of sperm are covered by sheath called manchet. Tail. The tail is several times longer than the head. Major part of it is called main piece, where the axial filament is surrounded by a thin layer of cytoplasm. The part behind the main piece is called end piece, which consists of naked filament. The sperm swims by the help of its tail in a fluid medium. Local course of spermatogenesis. As in the diagram, spermatogenesis waves moves in spirals like a corkscrew towards the inner part of the lumen. Outside on the edge of the tubule and at the beginning of the spiral lie the spermatogonia, and at the end of the spiral the fully developed sperm cells are in the lumen. From the diagram, it can be seen that several differing generations can be found in a tubule cross section. As time goes on, the wave of spermatogenesis is shifted towards the right in order to newly begin the process again. After spermiogenesis, sperm heads become embedded in the Sertoli cells or nurse cells. In the mature sperm, it consists of a long, slender bundle of filaments that propel the sperm by their undulating movement. Once the sperm has matured, they are finally released from the seminiferous tubules by the process called spermiation. It is transported through the long seminiferous tubules and stored in the epididymis of the testis until it is ready to leave the male body. Gonadotropin release hormone. Spermatogenesis is initiated due to increase in gonadotropin release hormone by the hypothalamus. GnRH acts on the anterior lobe of anterior pituitary gland to secrete luteinizing hormone and follicle stimulating hormone. Follicle stimulating hormone or FSH. The second hormone secreted by the anterior hypophysis is the FSH that affects Sertoli cells by triggering the formation of testosterone binding protein. Thereby, testosterone can be transported by Sertoli cells into the luminal compartment and concentrated. FSH acts on Sertoli cells to secrete an androgen binding protein and inhibin. ABP concentrates testosterone in the seminiferous tubules while inhibin suppresses FSH synthesis. FSH acts on spermatogonia to stimulate sperm's production. Testosterone. Between the seminal canals lie Leydig interstitial cells. These are endocrine cells that mainly produce testosterone, the male sexual hormone, and release it into the blood and other neighboring tissues. An initial active stage of these cells occurs during the embryonic development of the testes. Later, in juvenile life, due to influence of LH secreted by anterior pituitary gland, Leydig interstitial cells enter a second long-lasting stage of activity. Together with the hormones secreted by the adrenal cortex, testosterone initiates puberty. Testosterone is carried via blood and lymph to all the tissues, especially on the brain during development as well as on the sexual organs. It mainly affects spermatogenesis and the maturation of the sperm cells. During spermatogenesis, one spermatogonium produces 4 sperms. Sperms have half the number of chromosomes. After fertilization, the diploid chromosome number is restored in the zygote. This maintains the chromosome number of the species. During meiosis I, crossing over takes place, which brings about variation. Spermatogenesis occurs in various organisms. Thus, it supports the evidence of basic relationship of the organisms. Spermatogenesis is the origin and development of the sperm cells within the male reproductive organs, the testes. The testes are composed of numerous, thin, tightly coiled tubules known as the seminiferous tubules. The sperm cells are produced within the walls of the tubules of the testes. The seminiferous tubules are lined by germinal epithelium. The germinal epithelium consists largely of cuboidal, primary or primordial germ cells and contains certain tall somatic cells called Sertoli cells or nurse cells. Sertoli cells function to support and nourish the immature sperm cells by giving them nutrients and blood products. As the young germ cells grow, the Sertoli cells help to transport them from the outer surface of the seminiferous tubule to the central channel of the tubule. The process of spermatogenesis thus includes formation of spermatids and its development into spermatozoa.